Hi everyone, I hope you can hear me okay. I'm going to talk to you today about some Bayesian methodology. Some of you may recall last year I did a presentation on some Bayesian analysis that was used for a respiratory study, and this is a follow-up on that. So I'm going to do a recap of the methodology that we talked about last year and then talk a bit about how to analyze two endpoints together, which we had to do for this particular study. Okay, so the example study, we started off looking at one endpoint, which was the predicted FEV. So we want that to be high because that will indicate a better lung function. Two parallel groups, so active and placebo. They had repeated doses. So doses week 1, 4, 8, 12, and baseline was covariate in the study. 2 centres, and the primary endpoint was change from baseline at week 12. And one of the things that the client was interested in was the probability of there being a treatment effect more than 10%. So Sue actually simulated this data. And to start off, we did a simple ANOVA model, so looking at change from baseline in week 12. So we've got a proc mixed with treatment and centre as categorical variables, change from baseline, looking at treatment, adjusting for centre, and this is the baseline covariates. And we wanted to get the LS means out, and you can see the results. So the active was way higher than placebo with a difference of 9.88. And if you remember that probability we were interested in, which is calculated here, so the probability of this treatment difference of being more than 10 was just less than half. And then we were doing the same thing but using proc MCMC. So I'll just go through this code. So I know a lot of people haven't used MCMC very much, and it's quite an unstructured proc, so it's a bit like using, can't remember which one, sorry, but there's no structure, you kind of put in the model and everything yourself, so it's quite easy to get wrong because it will quite often run something which is just nonsense, so you have to look at the results quite carefully. So the first bit of the code, we've got the data set that we're using, selecting just the week 12 visit. Seeds random number, this is the number of iterations that you use, and the data set we want the results to come out of. Then we set up the model parameters. So B0 is the intercept, and B1 is the slope or treatment coefficient, and sigma is the variance. And then define priors for these parameters. So we're put B colon, that's going to cover any of the parameters that start with a B, so the slope and the intercept. And this is a non-informative prior, so it's a normal distribution with a mean of 0 and a large variance. The sigma has a different shape distribution, so I gamma, but again this is non-informative. Then we define the model, so this is like your y equals mx plus c. So we've got the intercept and the slope multiplied by the treatment variable, where treatment is I think it's a 1 and a 0 or a 1 and a 2. And so the change from baseline is going to be normally distributed, because this is the mean, and that's the basic model. If we want to add in the adjustment for baseline, we need to add another parameter. So this is a baseline parameter being added here. We don't need to add in the prior because it's already covered by this B colon. And then in the model, we've got the baseline parameter multiplied by its baseline variable. Now we're adding in centre. So centre, I've got like a dummy variable, so 1 and 0, depending on which of the two centres it is. So I've got the parameter centre coefficient, a prior for the centre, and then adding it in here. So this will switch on and off depending on whether it's centre 1 or centre 2. If you have more centres, you need to add in as a class variable for each of these centres, so it can get quite messy, which is why we've only got 2 in this example. That doesn't give you any output, so you need to add in what you want to see out. So if you're using 9.4, you need to add statistics equals all, otherwise it won't give you anything. And then you need to specify which parameters you want to monitor. So this is going to give you results and diagnostics. So I've put all of them in, but if you have a model that's got lots and lots of parameters, then this output can get really very large, and so it makes things very slow and clunky, in which case you want to think about which parameters you really want to monitor. The middle bit of the code hasn't changed, and then this bit here is really useful. So after you've specified your model, you can specify anything you want. So here is calculating equivalent to your LS mean. So the active will be the intercept multiplied by the treatment coefficient times the value for active, and then centre coefficient multiplied by half. So we're assuming half in each centre. I've not got baseline in here because baseline has been centred, so the mean baseline is 0, but otherwise you would need to add that in as well. Same for placebo, then the treatment difference, so those centres and the intercept cancel out, and the probability that that difference is more than 10. But you can add as many things in here as you want to. But whatever you define here, you need to add up in this monitor, otherwise it just won't show you anything for it. So I've got here the results of the ANOVA that I showed before, and then this is the same equivalent results but from the Bayesian. So as you can see, they're not identical, but they're quite close. So now moving on to new material, so looking at 2 endpoints together. So we've got the predicted FEV, same as before, and then CRP, and in this case a lower result is better, although it can get too low. So if you have co-primary endpoints with a traditional frequentist approach, it can make things quite tricky, because if both results are equally important, both endpoints equally important, you need to think about splitting alpha between the 2 of them and basically running 2 separate models. So it's quite frustrating if one of them is significant and the other one isn't. With the Bayesian approach, you can put both of those endpoints into the same model and analyse them together. And then we had decision criteria based on the 2 endpoints, which I'll come to later on, which was specified in the analysis plan, and then at the end of the study, decisions were made based on those criteria. So these are the criteria. So the first step was to look, as before, at the FEV being less than 10. And if the probability of that was pretty high, then that was unsuccessful, because we wanted to remember the difference to be more than 10. Now, if the probability of that FEV was more than 10 and the probability of the CRP difference is less than -1, if the probability of that was more than 0.25, then that was a really good, clear success. So both of the endpoints showing us doing what we want them to do, that was a robust success. If we had a pretty high probability, so 0.35 or above, if there being a difference in FEV of more than 10, but not looking at the CRP, then it was a success, but a kind of few, we're seeing something rather than party time. So the MCMC allows this analysis to be done together, and then you can get quite a funky plot coming out, which I think looks a bit like a fried egg. So here on this scale, the y-axis, we've got the FEV. So if you imagine it sort of 3D, you've got a normal distribution along here. So this is the peak, and then you've got the tails either side, and then the same, in fact, it's here. So if you, I don't know if you can picture it, but this is like the sideways view of this. If it was raised up. And then along here, you've got the same thing with CRP. So the most likely outcome, you're in this quadrant here. Ideally, we want it to be here, because that means that the FRV is more than 10 and the CRP is less than minus 0.1. And the probability, so we go through each step at a time. So the probability of the FEV being less than 10, so that's in this section here, is less than 0.75 because it was actually 0.1712. So therefore, the first step is not potentially unsuccessful. The second step is to say what's the probability of it being more than 10 and also the CRP being less than minus 0.01. So that's in this section here. And the probability of that is 0.2962, so more than 0.25. So therefore, in this example of simulated data, the result is a robust success. So it's all good. But I think, although the analysis is quite complicated, if you imagine you're trying to describe the results to a study team, clinicians, this is quite a nice representation of the results. It takes a bit of time to get your head around, but seeing the two endpoints together, I think, is quite good, quite helpful. So the results meet the criteria for robust success, and the joint modelling approach with the Bayesian posterior probabilities makes it easier to interpret the results. So the methodology, I won't get too heavily into the stats, but we're assuming that the two endpoints follow a multivariate normal distribution. So this is a bit like the diagram of the posteriors, really. The way that we modelled it, we had each treatment arm had a separate variance-covariance matrix, and I'll hopefully show you the SAS code for that. No intercept terms were added, and the baseline was added as a continuous covariate. It was centred, as I mentioned before, so the mean was 0. We just made it a little bit easier to code. Non-informative priors were utilised for each of the model parameters. So the placebo, active, and baseline parameters had a multivariate normal prior distribution, so mean of 0 and large variance. So quite similar to the single endpoint. Okay, this is where it gets a bit ugly, but you go through the code a little bit of how to set up the multivariate normal priors, because it's not completely straightforward. So this would be like the mean, and it has two elements, so one for each of the endpoints. And then we set up a 0 matrix, which is just going to be a temporary matrix, which has 0 for the two elements. Sigma is going to take the form of a 2 by 2 matrix, so this is the variance-covariance matrix. And then, because it makes it easier to code, we've got a temporary one as well. Fill matrix, what this does is fills all the matrix of the temporary matrix with the value that you're specifying. So we're going to get a 2 by 2 matrix with all 4 cells having this, which is going to be the variance, a really big number. And then if you remember from your college days, the identity matrix is going to be that, we're calling that sigma, so that gives you the diagonals of 1s and 0s for the rest of the cells. Then what this does is it multiplies the temporary matrix, so the one that's got this value for each of the cells, with your identity matrix, and it's going to replace sigma 0 with the result. So it gives you this basically. So this is going to be your prior for the placebo. So the mean of 0, and that's your sigma. So it's quite a lot of code to produce something quite simple at the end. So the same will be done for the, oh sorry, no, I'm just skipping ahead. So the variance-covariance matrix for placebo and active will use its inverse Wishart distribution. So this again It's non-informative, we hope, but it's a little bit more tricky to find a non-informative for this distribution. But we need to use this inverse Wishart because it makes the maths easier, makes the posterior probabilities come out as normals, which is what we need. There's a dependence between the variance and the correlation. So if the correlation is exaggerated, then the variance is going to be higher. So you need to watch out for this, because in this case, this is our posterior, and that's our data. And there's quite a good match between the two of them, like they line up, but which indicates that the prior is non-informative. But in some cases, if the variance is quite large, you might find that you need to look at the prior and change it because it's not truly non-informative. So this is the joint code. I'm not going to go through every line of it because it's quite long. I mentioned in the slides that we were fitting separate variance-covariance matrix for the two different treatments. To do that, we had to split up the data set so that we had a different variable for the placebo and the active. So basically said if the treatment is this, then we created two different variables, so one for active and one for placebo, rather than having them in the same variable, but just with a treatment variable to indicate which was which. The other thing is I'm only modelling week 12, but if you're doing repeated measures, you need a wide structured data set, so with the time points going along, whereas for something like PropMix, you use a vertical structure. So we've got MCMC code here. As you can see, we've got all of the parameters that we want to monitor, like I did before. This is setting up all the different arrays. So here are the outcomes, so the data that we're modelling. So the first, so data 1 is your placebo data, so for the FRV and the CRP. Data 2 is your active for the FEV and the CRP. We've got two baselines, so again, because we've got two endpoints, two baselines, FEV and CRP. This is just like something that's a linear predictor to do with the baseline, so I'll show you where that comes in later. Then we're going to calculate the treatment differences, so we need a parameter for treatment difference. So again, there's two, so we need an array for this, whereas in the previous code we didn't need these arrays. This is going to give us the probabilities for the different quadrants. If you remember from that plot, we had a probability for each quadrant. These are going to be like the LS means for treatment and active, and baseline covariance, sigma for placebo and active. This part here is setting up the priors. So this is the code that I showed you before with the zero matrix and the identity matrix and everything. So this is setting up the priors for the sigmas. This here continues, so we've got priors for sigma, we've got priors for the means. Now you can, the parameters here, you can put starting values. And I have found that sometimes these are quite important. So if you put just anything or leave it blank, if you leave it blank, it will set a starting value of 0. If you put in a value, it will use obviously the value that you give. If you give it a silly value, you can get some really crazy answers. It shouldn't really make a difference in an ideal world, but I found that it did make a difference. So that's something to watch if you get numbers that don't seem very sensible. So we've got priors for the treatments, priors for the sigmas and baseline. And then this is the model statement. So we've got it in a do loop because of the two endpoints. So it's got baseline and then the treatment differences, the treatment for placebo and active. Haven't put center in here because I was trying to keep it simple. And then this is similar to what we had before. So placebo is going to be normally distributed around this, and then the same for active. This is the data that's coming out, and then here are all the calculations that we want to see. So I've got treatment difference, and then here are those probabilities. So the probability of both being successful, both failing, and so on. Underneath that, we've just got a prop mix, just a sense check. And that's really useful because, as I said before, you can have MCMC run and it all looks like it's working, but because you set up the model statement wrong, you get crazy answers. So it's really good to have something just as a double check that you've got things roughly as you were expecting. So it's really important to look at the diagnostics. For the study that we use this methodology for, we've got some really crazy diagnostics out. So it's really important to check them. I'll show some examples in a few minutes. It's also quite useful to plot the posterior distribution for the key parameters so that you've got an idea of whether the prior is informative or not. And we reported the posterior median, the standard deviation, and the credible intervals, which are like confidence intervals. So one statistic that came out as a diagnostic, which is really useful, is this ratio of the MCSD to the SD, standard deviation. So this is basically showing you how much of the error is due to variability in the data and how much to do with the model simulation error. And it should be ideally less than 0.01, although if that's really difficult to achieve, then 0.05 is reasonable. To make it smaller, you increase your number of iterations and increase the burn-in. So the burn-in is how many iterations you want to discard at the start. This here, this diagnostic compares the start of the chain with the end. So if you've got a large Z score, it indicates poor conversions because it means that the start of the chain and the end of the chain are quite different. So here's an example of really disastrous diagnostic. So we've got quite a small number of iterations here and no burn-in. And this, as you can see, it's like bouncing along all over the place and it looks a bit of a mess. And the MCSD to SD ratio here is very high. So if you've got that, I mean, it's just horrible. So either you haven't got enough runs or there's something really wrong in your model. You can see that increasing the number of iterations tenfold has made it look a lot more respectable. It's not perfect, but we had some that looked like that and we thought it was possible. It kind of depends on your data, how many data points you've got and how nice it looks. Here it's better still, so we've got 50,000. This has come right down to 0.02. The burn-in on this one is not really very clear, and sometimes you get something really different happening at the start of the chain before it settles down into this pattern, in which case you would want to increase your burn-in to get rid of those where it's sort of doing something funky at the start. And then here it looks, you know, really quite a good hairy caterpillar. It's got a very small ratio here, and this autocorrelation that drops down straight away, so this is kind of very healthy looking diagnostic. Having said that, you get this, it still doesn't mean your estimates make sense, so you need to check those against Endpoint mixed as well. Now, this was the output I did from the ANOVA, and you can see a treatment difference of 11, and then here minus 0.06. We didn't get a brilliant match actually from the MCMC. It's quite different results, and I didn't have time to look at why that was. But because this was the simulated data, I didn't spend too long worrying about it, to be honest. So if you were doing these analyses for a study, one thing you might want to look at is what would happen if you change your priors. Would it change the outcome of the study? Because if so, there's something maybe wrong with the priors. So we didn't really look at it in too much detail because for the study, basically the results were a disaster, and it showed the study didn't work, so we didn't spend a lot of time, and the proc mix showed the same, so we didn't spend a lot of time worrying about the priors. But that is something to be careful of if you are going to use the results for something. So I found this methodology really complicated to use, very time consuming. It's also really difficult to QC because you don't get exact match. Like the QC and the production side, not going to get exactly the same numbers. So you have to go through manually and check whether you think the numbers are within an acceptable range. But the plus side of it is you get this really nice way of interpreting the results, which is easier for a clinical team to understand and easier for them to make decisions. You need to be careful about choosing priors, and we had to tweak the model quite a lot to get it to work. So if you're writing a RAP, you need to leave a bit of flexibility in the RAP to say, different elements of the model are going to be investigated so that you're not tied down too much. And very, very time consuming. Some of these models took sort of 4 or 5 hours to run. So it's quite an undertaking from that point of view. Okay, that was all. Does anyone have any questions?